Hi, thanks ever so much for the invitation to present at this meeting. I'm Hannah Batchelor, working at the University of Strathclyde, and my presentation today is going to look at the formulation considerations for bioavailability and bioequivalence studies for paediatrics. What I wanted to cover firstly was a bit about when the paediatric formulation is considered in the standard development pathway. I've got a further slide on this, but I firstly wanted to put this into the regulatory context. Across the top of this slide, I've got the typical adult programme pathway where we go between phase 1, phase 2 and phase 3. And at each point, we've got relative fit for purpose formulations that exist at those time points. You can see from this that the EMA require that the paediatric formulation is considered at the end of phase 1, whilst in the FDA, it's after phase 2. These two different time points can mean different things about the paediatric formulation relative to the adult, and it's worth considering how this is managed during development, particularly where products are likely to be global and thus need to meet the criteria of both the EMA and the FDA. This puts the previous slide into a little bit more context, where here I'm looking at the bridging for an adult to a paediatric formulation in the greatest overview. At the top again, you can see the standard adult development pathway, where you've got enabling formulations at the beginning phase to the marketed formulations towards the end. Typically, the commercial formulation is used in the phase 3 studies, if that's possible, to avoid bridging right at the end of development. For the paediatrics, you might have the same preclinical package, but it doesn't follow the same phase 1, 2, 3 as for the adults. It's often driven by the adult development, where the paediatric formulation is brought in later in the pathway to minimize the burden to paediatric patients. The way that people bridge is highlighted on this slide by using things like the in vitro and in silico packages, using relevant biomedia as biopredictive techniques to look at the effect as to how the paediatric formulation may differ from the adult, as well as relevant animal models. There's also some relative bioavailability studies that might be conducted between the existing adult formulation and the paediatric formulation to highlight any differences. And those two packages of work are used to inform the study that we conducted in the paediatric population to demonstrate the dose finding, safety and efficacy prior to having a marketed product. And again, typically the final formulation, or as near as, is used in all paediatric studies to avoid bridging further down the line. There's no clear guidance on how to manage this bridging, and this is a big issue at the moment because everybody is using slightly different techniques, and there's a need to bring this together so we can understand the best ways to bridge from adults to paediatrics, and even more to understand where there's specific risks involved for particular products or drugs. In terms of these relative bioavailability studies, there is no stringent guidance, but I've just summarised here the three major ones. So the ICH, the FDA and the EMA, with the extracted sentences on what they consider to be appropriate for relative bioavailability studies between paediatric and adult formulations. And from what I've highlighted on the slide is it's typically recommended that it's conducted in adults. So we're using adults to compare the adult to the paediatric formulation, and then putting that paediatric formulation into a paediatric population without having tested there previously. And I'm going to talk in this presentation about some of those risks and how we mitigate them. So this is just a sort of schematic really about the factors that affect bioavailability and why this might be different for a paediatric versus an adult product. So in this slide, you can see there are various drug dosage form effects, things like the excipients. Now the drug substance is going to be the same. So the drug substance, the amorphous or crystalline form, solubility and intrinsic dissolution rate are likely to be similar, assuming the same form is used between the adult and paediatric product, but the excipients may change and the manufacturing process may change. That will all affect the dissolution properties. Once the drug is dissolved, the absorption should be similar in the same population, but then there are differences between paediatrics and adult populations. So I've just put these summaries here. So technological factors might have a similar influence in adults and children. However, you need to consider the dissolution rate. Dissolution is a function of the drug that's dissolving in the solvent available. If there's a change in composition or volume, then the dissolution will be affected. There's also the impact of the physiological factors that might be different in children to adults, and I'm going to speak more about that later in this presentation. I've just put here a range of paediatric oral formulations, mainly to highlight that where possible, for oral products, an immediate release film-coated tablet is typically the product of choice for an adult, whereas for children, there's a range of products from granules to chewable tablets. Still liquids are popular, and generally smaller units. Swallowability can be an issue in paediatrics. So where we're making a smaller unit that's simple to swallow, we have removed the disintegration step that is present for the adult formulation. So if disintegration was a rate limiting step, then you might expect a change in performance. I'm not going to go into huge detail about the key physiological risks about paediatrics and adults. I know there's other talks on the programme that might cover this, but it's really just a highlight, and I'm specifically going to talk on this slide on absorption. So here we can see there could be changes in gastric emptying, surface area, transit time, and the impact of food. These are the key ones that we're interested in for the relative bioavailability and bioequivalence of an alternative formulation. So I'm presenting here on this slide a case study to show that this is a real risk that we should be considering. So for lamivudine, there was an ARROW clinical trial where it was exploring pharmacokinetic data in children who were switching from a solution to a tablet. And as you can see there in the bullet points, the bioavailability following the solution was lower in children compared to the tablet, whereas in adults, these two formulations were considered to be bioequivalent. So this highlights when we're doing our relative bioavailability and our risk assessment using an adult population, there are times where this doesn't extrapolate to children. This is really the same information, but just presented on a slide here to bring together everything that I've said so far. The bottom pictures are showing pharmacokinetic data. So for the orange and the black product showing equivalence in our two adult populations, whereas in the paediatric, when we go from adult to paediatric, we don't know whether the products are still going to be bioequivalent or not. So there are some risks about doing the studies in adults, even though that is still the preferred methodology proposed by the ICH, the FDA and the EMA. So that's a summary of everything that we've thought about to date. And I was discussing this topic in the AAPS meeting a few years ago, where we got together with some FDA people and put together a project where we really wanted to explore what our degree of certainty that differences in absorption from the formulation of the paediatric patients are correctly detected in the healthy adult volunteer study. Now, this project was funded by the FDA Generics division, so it's very much looking at generic products where the bioequivalence studies are conducted in adults. The product is introduced and quite often it's used in paediatric populations. There's a specific driver from them to explore this topic. Because of time, I'm not going to go into huge detail on this presentation, but I'm going to give you a whistle-stop sort of a flying summary of it. So the key goals of this project was really, they wanted to identify the generic products that are most at risk of suboptimal efficacy. And we were going to use the literature to scope evidence. And also, once we had found example case studies where there was differences in the bioavailability in children or in children compared to adults, we were then going to conduct some in vitro, some in silico modelling to see whether those models would have predicted those changes, and if yes, how we can then implement those going forwards for products that might be at risk. We first had to scope the literature to find evidence of these products. So we identified 40 studies that had bioinequivalence results or different relative bioavailability. And then a whole lot of information was extracted from each of these studies. We were going to create a database in this information that we could then mine to really unpick the reasons for bioinequivalence and choose our most relevant case studies. Just to highlight on here that the major thing that's of interest in this presentation was that we tried to identify from the literature the source or the reason for the bioinequivalence that was observed. And for this, we extracted the information that had been proposed by the authors in the original documents that we reviewed. This slide here is showing a summary of those results, and we themed all of the reasons rather than just giving you a list of however many there were. We themed them to things like the physiological factors, drug substance or formulation, the disease, population or study design. You'll note that the numbers of the studies identified adds up to more than the 40 that we explored. And the reason for that is often there was more than one reason given for the bioinequivalence, so they could be counted more than once in this table. You can see from the table and the figure is just a different way of showing the data, that the age-related absorption effects were presented in 27 of our studies identified. So that's quite a high proportion and worth exploring going forwards. There was also a lot to do with the inter and intra-individual variability. Again, paediatric studies are conducted in patients rather than healthy volunteers, so that may not be a surprise. For an example here, this was just one of the reasons that was suggested. The capsule formulation was providing higher exposure compared to a solution in children. And the reason that the author stated was due to the gastrointestinal time of the solution being shorter. So there was a more rapid transit in children, meaning that the overall absorption was reduced. This is the kind of information we were extracting. I just wanted to give a couple of examples so you can see where the data came from. A second example from a different paper was where the solution may lead to very high concentrations of active components, resulting in the saturation of membrane transporters. So the fact that it was already pre-dissolved in solution can saturate the transporters, which wouldn't be the case for the tablet where there's a slower rate of dissolution. These were interesting to pull together, and it was really a good exercise to explore the literature to understand more about these inequivalences that were observed. So I guess it leads us to what should be considered when we're predicting in vivo performance. How can we make sure in the future that we are not getting these differences? We're not having to explain differences in bioequivalence. How can we make sure that this is consistent throughout? And this is just a schematic really, where we're bridging between the clinical dose, the enabling dosage form, a pivotal dosage form, and potentially a commercial product. We've really got to think about whether we're looking at the adult form or a pediatric variation, the dosing and the increments, and how we're exploring these different aspects. We're really trying to make sure that we're getting the equivalent to the pivotal formulation in terms of the matched exposure for children versus adults. So we're bridging back from this commercial to make sure that this is always being consistent. Modelling and prediction, or modelling and simulation, is one aspect, and this can be very useful to bridge at both stages during the development pathway, but it does have cost implications. A higher cost version and a potentially riskier version, because you're leaving it till later in the day, could be clinical studies at different phases. So it's really important to consider the risks associated at each bridging strategy and think what we need to do for our paediatric formulation as opposed to our adult. There's also a need to consider study designs versus real life. So for adult bioequivalence or relative bioavailability studies, there's typically quite stringent conditions. So we're normally using healthy adults as a start in the fasted state, or after a very standardized meal. It's all given at a very exact time with 240 milliliters of water, a measured dose with often exclusion factors relating to tobacco, contraceptives, and other drugs. In children, the situation can be different. Because it's patients, it's really hard to control how we dose and how that maps over. A 10-hour fast would be quite tricky to implement in pediatrics in real life. There's further in vivo performance considerations, and it's really to look at that adult data to determine the best starting point. What do we really know about the formulation from clinical testing, and what are we confident about extrapolating, and what aren't we confident about? Do we need multiple dosage forms? Pediatric spans from neonates to adolescents. Is it realistic that there'll just be one pediatric formulation variant, or will there be more? And are there any distinct cohorts within your pediatric population? The other thing that you might want to consider is that are there other special populations that might benefit from this dosage form? So could we use the pediatric dosage form and then use that for every participant? Is that feasible? I've just put together some summary conclusion and further thoughts. Like I say, this was a really lot of information packed into a short time, so I'm really happy to present this, and I'm hoping there'll be questions. But we're really using the prediction of relative bioavailability or bioequivalence to support formulation development. So should we be creating the formulation and proving bioequivalence, or using that data to inform our formulation? And I think there's a mix of both. We've got to really look at that in different age groups to support the dosing that's required. And ultimately, our pharmacokinetic studies conducted in paediatrics are really there to show how we can get our dosage regimen to achieve the same systematic exposure, which is safe and efficacious. That's a real whistle-stop tour, but I'm more than happy to be here, and I invite your questions. Thanks very much.